Hello and welcome back to Jono's Lab. In today's video, I thought I'd share my journey so far diving into genetic engineering of bacteria, trying to fill out my DIY biotech tree and build up some skills. And specifically the project that I've been playing with over the last number of months now has been trying to engineer a visible green E. coli strain that I can use for agar art. So as some background, my friend Sebastian sells a set of chromoproteins. So these are bacteria that are expressing these colorful protein structures. And he's got pretty much the full rainbow. You can see they look lovely, and they also, some of them glow under UV. So the green and red here I used along with a blue glowy bacteria that I found in the environment to do some agar art, and that's all wonderful. But in visible light, the green isn't really green. It's kind of like a yellowish. Sometimes if you grow it out long enough, you can see a sort of lime greeniness to it. But I thought it would be really cool if we could engineer something more obviously green, like a nice forest green. And so that was the kind of goal from the start, was could we do this? And so the first idea that I had was, what if we linked a blue and a yellow chromoprotein together? And so I designed this construct that was a yellow protein and a blue protein just linked by some long flexible linker. And then I realized that the blue protein and the yellow protein had chosen, in nature, one occurs as a dimer. So that's two of these units bound together, and one occurs as a tetramer. And so the two would probably be incompatible and fold nastily and interfere with each other. So I abandoned that in favor of just expressing both proteins individually. But it turns out Seb had actually tried this way back in the day, in 2022, and he'd managed to get it going, although he noticed that growing it out on plates, the color wasn't really visible. You had to grow it in liquid culture for a very long time, let it mature, and then you could see this nice green. So only when the cells were spun down and it had ages for that protein to accumulate. But he managed to get the color, so that was encouraging. I only found this out way after I'd finished my attempt. But speaking of my attempt, in my case, it didn't so much turn green as blue. And even the blue took ages to accumulate. In fact, I thought that thing had completely failed. It was only when I went to properly dispose of the plate on which the bacteria had been growing after like a week or two of sitting that the blue color was visible. So a bit of a sad failure for V1, but also this is biology, everything's messy and unexpected. My consolation prize was that I had modified the amino acid of the yellow protein to include the final additional letters GREN. So I technically made green, even if the actual result was blue. But it's still cool, right? We could take the genes for this blue protein that's originally made by a sea anemone, and we could insert it into bacteria, and we could grow that out and see that there's a change. So at least we had some of the pieces working. So after that sort of mostly failure, I stewed on this for a while. I'd done some research into other ways some different animals try to achieve green. It's kind of surprisingly unusual to see much green pigmentation in nature. A lot of greens are structural, which is not so easy with bacteria. But then my favorite email of the quarter, the Southern African National Spider Association, Spider Club of Southern Africa, an organization that I used to be part of, and I know a bunch of the people there. They send out this big sort of amalgamation of all of their research in a quarterly newsletter. And they had a lovely article on crab color spiders. So Prof. Dippenar Struman, I've actually worked with her on some crab spider stuff. But anyway, she'd done this lovely survey, and they had a section on the green coloration in some of these spiders. Structural coloration is common, but also there is some pigment-based coloration based on biliverdin. And this is something I'd seen looking at other insect greens as well. But this article was like, okay, it finally kicked me into that's probably the one I want to try as a second attempt, right? And that gives this lovely green color in the spiders. And the way this works is that biliverdin is a single step away, in essence, from heme. So this is the heme molecule. This is, the heme from blood, but it's also found in many other, it's pretty much every living cell, including bacteria, albeit in small amounts. And so the plan was to express this protein called HO1, which is heme oxidase, right? It takes this heme, it binds it in its active site, and then it's doing this transformation, this breakdown of splitting the heme into the biliverdin. And so I found this is a well-characterized protein. There are various variants. I found one codon optimized for bacteria. And so my next design used that as the basis, expressing that HO1 gene and hoping that we'd produce biliverdin. Unfortunately, the colonies themselves just look basically the same as normal, pale white colonies. But if you looked at the plates from the side, you'll notice this bottom one here is the one that had the modified bacteria growing on it, in contrast to the top one. And you can see there's this dark greeny brown, almost bruise-colored coloration. And by the way, I say bruise-colored. Bruises in your body are a lot of the pigmentation there is from biliverdin, and then a further oxidation product of that, bilirubin, and others sort of continues to oxidize further into these browns and yellows. If you've ever wondered why your bruises look lovely, greeny brown. But anyway, that was the color of my agar, but not of my bacteria colonies. So a little bit disappointing, but also not entirely unexpected, because this is a bit of a hopeful step. I actually knew that I'd probably need more than this. And from the start, I'd planned on needing to do additional steps, but I thought rather than trying to do too much at once, I should at least begin by seeing, can we even express and produce biliverdin in usable quantities? And it did take a while. It took longer than I'd like for even this color to show up. Like I said, the colonies themselves didn't look colored at all. But it was still useful to know that at least the enzyme is being created and it is working. It's just that the small molecule is not necessarily having any incentive to stick around in the cell. We don't have any machinery to, put this in a vesicle or something. And so it was just diffusing out and spreading and diffusing throughout the whole plate. So how do we solve that? Well, there's a few different ways. The one that I liked was based on some further research that I came across. Multiple origins of green coloration in frogs mediated by a novel Biliverdin-Binding Serpin. Right, so this is, it turns out, a whole class of solutions that have evolved multiple times in these frogs based on a sort of commonly used backbone type of protein called a serpin. It's a whole class of proteins, but they've been adapted to bind to the pigment biliverdin and also to modulate how it absorbs light and what color it looks like as a result, right? So this more yellowish, brownish green becomes this almost like bluish cyan green in some of these frogs. And so, yeah, the paper was fantastic. They studied how this protein is binding to the biliverdin, preventing it from further oxidizing, localizing it, but also changing its absorbance, and then ending up in the various fluids of the frog and contributing somewhat to the overall green color. And they did some cool analysis and so on. I think there's been follow-up works on what exactly is happening to that molecule as it binds to that protein. And so, yeah, there've been some little studies of even putting one variant of this into E. coli, although I didn't want to copy that work. Instead, I picked purely on aesthetics, the particular frog that I wanted to crib from. And again, it's a protein that we're producing with a binding site that's going to bind to that biliverdin molecule, hold it in place. And hopefully then, instead of it being a tiny molecule that can just diffuse out, it's now linked with this much larger protein. Now in the frogs, the protein also has a peptide sequence that kind of tags it for secretion because they produce it in the cells and then secrete it into the fluid. But I clipped off that tag in this case so that you just have the main protein and it's not hopefully going anywhere particularly fast and should stay within the cells. So once again, I created my construct with all the pieces, and we can talk a little bit about how that works just now. And I put it into some E. coli. Unfortunately, it didn't really go greenish. I mean, if you enhance the saturation, right, and squint charitably at this, this is not white. This is definitely greenish, right? So we can claim some sort of victory, but it took a while for that color to develop. It's still, this is much closer to what it looks like to the human eye. It's kind of like a pale yellow. There's definitely still some biliverdin diffusing out rather than sticking around in the cells. Yeah, so not quite the final outcome we're waiting for, but probably where I'm going to leave it. So to debug why, there's a few things that I still maybe did not ideally in this. One is basing all of this assumption on the availability of heme as the sort of initial product that we're then going to break down. Right, because heme is not super abundant in E. coli, and if you are using up all of the heme, you're also kind of interfering with its metabolism in general. So there's a strong pressure away from retaining your plasmid because it's using up this like kind of important molecule. So if you really wanted to do this well, you'd probably upregulate some of the genes that code for the precursors of heme as well, and maybe even supplement your agar with something like alanine, right, or just iron even, to try and boost the start of the chain, because it's actually a whole chain that eventually leads to heme and then to biliverdin. And then I actually tried this. I crushed up some iron supplement pill from a friend, and you can see this gradient of dissolved iron. The pill itself has some coloration as well, so don't read too much into this, but definitely the colonies that were nearby where there is some extra iron seem to have slightly more visible color. But also the other problem that I made, and the larger mistake, is that when I was ordering this plasmid, usually you have this structure where the plasmid comes in a plasmid is a ring of DNA that we're going to use to deliver our genes that we care about. But it also codes for resistance to an antibiotic so that you can select for that plasmid. It codes for some replication machinery that tells the cells to make copies of that plasmid so that they're passed on to the future generations of cells and they stick around. And then you have your main piece where your DNA goes. And now there's different backbones that the company that I ordered from GenScript provide. PUC57 is a cloning backbone. So usually what you would do is you would use this to make lots and lots of copies of your DNA, and then you kind of cut and paste out the piece that you cared about and insert that into some different vector later. They also have more expression-focused ones that can be turned on and off. But in this case, I wanted an always-on pigment gene, right? So that as soon as you grow the bacteria, it's colorful. I didn't want to be messing with induction. And so I have my own promoter and ribosome binding site, RBS, and then my own terminator kind of in this insertion cassette here. So when I was ordering the this third and final design, I forgot to change away from the default vector, which is a very cloning focused ampicillin resistant backbone, as opposed to the kanamycin resistance version that I've been aiming for. Anyway, so luckily I had a tiny bit of old ampicillin, enough to make a few plates, but it wasn't quite strong enough. You can see it's degraded, and so as a result, in the middle of each of these dots is one colony that was successfully transformed with the DNA. But then as soon as that started producing the enzyme that's giving it the antibiotic resistance, a lot of other bacteria that don't necessarily have the genes that we want were able to also survive next to those colonies. So we call these sap-like colonies. So my transformations of this into a bacteria that would be better at producing protein were kind of foiled by this satellite colony dynamic, and just in general by, I think, the design not being particularly wonderful and my ampicillin being old. So if I were to do this again, I think the main things would be, maybe supplementing the agar with alanine or with some precursor to heme to try and boost that whole process. Maybe looking at including a copy of a gene that would be helping produce that precursor, but also more importantly, experimenting with the promoter and the backbone to try and figure out something that's going to be higher expression of the two proteins here, the HO1 that's producing the biliverdin and the Biliverdin-Binding Serpin. So those would be the main changes for if I was to do another attempt. But I think I'm probably going to call it here. I've learned a lot about metabolic engineering, mostly how hard it is, a lot about the whole process of designing and ordering these, done lots of transformations, good practice for actually getting the genes in. So I think that's my overview of this particular project. If you have questions or curiosities, do leave them in the comments. I'll try and put references to like the main papers that I found of interest. And also I have these design files public on GitHub, so if you want to see how I assembled the cassette of DNA for the promoter and the gene that we want and the Biliverdin-Binding Serpin, how we make sure that these parts are going to work out, how we double check things. I'll share this code. This is just finding those sequences, stacking them together, converting between the amino acid sequence and the DNA, assembling them together. This was my planned target vector. What ended up happening was, like I said, just leaving it on the default by mistake when I ordered. Another piece, these Biliverdin-Binding serpins are glycoproteins. So in the frog and in eukaryotes in general, there's some extra machinery that modifies the protein, which the bacteria don't necessarily have. I did find a paper that had found that they worked okay even without that, so that's one of the reasons I kind of plotted ahead. But still, it's like this is a frog-optimized protein being injected into a bacteria, which at the end of the day is is not necessarily going to be ideal. But anyway, we can still assemble that together into this big string of DNA, have that synthesized, and like I said, it did seem to make at least some vague color, albeit not the beautiful dense green that I'm hoping and dreaming of. Cool. I hope you found that interesting. Thank you so much for watching, and we'll see you in the next one.